Thanks, Kevin. And if I could bring Deirdre and then also introduce Stephen and Thomas from Aracari and CN Bio, respectively. Okay, just to get us started, first question to Kevin. So how should we go about forming this test list of compounds? You're going to be publishing something soon? Yeah.
We're going to publish it soon, and we will have justification for each of the drugs that we select and also for the ones that we drop. So there are various, various data sources that are informing us on that, such as FAIRS, literature, reactivity potential, physchem drivers, molecular descriptors. There's a lot of information there. So don't worry, we will have everything clearly justified.
And so I guess to the two model developers on the stage, what are your thoughts on the preclinical, non-clinical species debate here? Because Danny this morning clearly said that it's a lot of work to develop an animal model de novo, but there might be regulatory needs. So where do you see the optimal path forward there?
Yeah, maybe I'll jump in. I think in short, I'll support what Daniel started this morning. Building one model is hard. Building 4 or 5 or 3 or 4 is, exponentially harder. I think one small example would be getting hepatocytes from multiple species is probably possible, but getting the non-parenchymal cells is then another level. And I think if we're going to have these models with the capability that I think everybody in this room wants, actually, that was listed, I think, very nicely by Deirdre here, that actually we need to have that, and actually that's a real challenge. And I think that the human models have got well advanced. Actually, there's a lot of things we could do with them in a relatively short period of time. All the things that are being described, start to compare them with a level of commonality, while potentially some of those non-preclinical animal species are almost developed in parallel, and maybe come along in a future phase.
Kevin, do you have thoughts from a regulatory perspective on the non-clinical species? Do you see that it's necessary from your perspective for a smaller, narrower context of use, or do you think that we will need them for?
I would think so. Perhaps for investigating, species-specific finding. I think that might be important. Or if you see something very odd in the rat, if you want to assess translatability to human, I think that would be quite useful. It's kind of case by case.
I think the only thing I'd add as well is that hasn't really been discussed is also about mechanism, right? So I think if we can get to a scenario where you understand why something's been toxic in a rat, say, but not in a dog, if you can recapitulate that mechanism in your MPS, human MPS, well, then you can have confidence that the two are going to translate. So bringing in those omics type analytics, like that wasn't actually on any of the slides, but we can do full omics analysis relatively easily these days on a chip or on a rat, and actually then you could get the translation between, well, what mechanism is actually causing the tox. And that might be one way in the short term to bridge that issue, rather than having the thing that maybe we all kind of want, where you have all the species and all the setups allow you to do the kind of perfect experiment, which hopefully everyone can see is maybe not where we are today.
Can I say something? So I definitely agree with you. I think, I'm a big omics person, and I would say I totally understand that, with spatial omics and, those types of technologies now being able to, come out. I had some slides, I took them out of that to make it shorter, but I would say, in line with that, and in line with, I would say, animal use, what typically happens is where I think that the complex in vitro models and understanding that can help with understanding the mechanism is that, most of the time, I would say, you do screening and, may see something, may not see something, but a lot of the times it's those longer term studies that you're doing. So you do a 7-day study, you do, with 1 month study, and you get, additional findings there. And you spent all that time, you have those studies, they could have a month recovery, understanding and even seeing that pathology data from there to understand what you have. I mean, that's, could be 6 months from the time you started. And, just, going back and then trying to take that and then, go redesign animal studies, use more animals, try to really understand and hypothesize what could be the mechanism. Having a complex in vitro model that has those cells will allow you to do that so much faster. And in the end, be able to get the patient sooner. Because, or if you see something in the clinic, really backtracking and translating what is the mechanism in that complex in vitro model, how does it, line up to, what we saw in the animal? Why didn't we see it in the animals? Like, what's different about it? And, it's just, various different ways you can look at it. I think, they all have their merit as to, I can see both sides. But I would say from that standpoint of developing, models, it's a lot easier to be able to utilize and understand mechanism using an in vitro system.
Nick, is there a?
Okay, now it's on. All right, so this is a question from our attendees online. And so it is, how important is it to match clinical endpoints such as AST, ALT, et cetera, to the CIVM readouts? As an example, decrease in albumin has been a very consistent indicator of toxicity compared to release of AST. However, in the clinic, decreased albumin is only seen in end-stage liver failure. Will surrogate measurements be more difficult to validate compared to those traditionally used to show liver injury in the clinic when using a CIVM? And what are your perspectives there?
Maybe I'll go first. I think as much as possible, we've got to bridge that gap, right? We've got to have as many of those endpoints and biomarkers the same and correlated, but there's always going to be some that are not going to align. Depending on the limitations of the model, you might not have every cell type. Some of those biomarkers are getting driven by inter-organ crosstalk. And so there are going to be limitations, but I think if we can, and what we're starting to get to in this group is having a cohesive set of biomarkers that we can kind of agree on. I think that there will then become a majority of key ones that can be used sort of more broadly and more consistently. And they obviously really need to be the translational ones. That's definitely key.
Thanks so much for the talks and the panel discussion here. I wanted to ask a question about the need for animal-specific chips, but then tie back to the compound selection, drug selection that Kevin was touching on. So, and I think people can appreciate at a place like FDA with a lot of people work there. There's going to be differences of opinion, within a regulatory agency there. And one thing that I always found interesting from like workshops is then sometimes you can hear differences of opinion from different people, like at FDA. And I wanted to ask sort of the panel's thoughts on, so like if you see a difference between species, like one possibility is or a difference between an animal and a human, let's say, is that, it had something to do with the actual liver cells. But another possibility is there was something else about the experiment, like there was higher exposure. I'd be willing to bet a lot of money that's like what drives it most of the time. And like or length of exposure and like clinical pharmacology factors separate from or like pharmacokinetics, there's like a whole nother part of it. And like just getting a dog cell versus a human or rat, that's like one small piece of the puzzle. So it'd be interesting to hear any perspectives on that. And then the sort of second part is like Kevin spoke about, rationale for why we might want to select other drugs as like reference drugs. And I'll add from like the experience in the cardiac space, like this is the probably the most challenging part, and like what was criticized the most for years. And in the end, it comes back to the context of use. So like, there's no magic list that like, this is the list of 20 drugs. I would just view it as like, these are some considerations, and you don't necessarily have to have a really long list if you understand like the context of use is this mechanism, and or you understand the other principles. There's other ways to get at validation potentially, but I'd be interested in perspectives on the, let's say if you're looking at a context of use of, you have a difference between animal species, and I mean, do you really need a animal specific chip or not? I think there would be differences of opinion there.
I think to start out is kind of focusing, I think what a big part of Kevin's talk was thinking about like the standardization of these models and how we perform our experimentation and all these tests and trials and validation that we want to do on them, to really make sure that that the data that we're getting out of it is, meeting our goals and our hypotheses, but also that all the trials that we do across, whether it is a human cell model or animal cell models, that those cells across all of our different sample sizes and all of our different experiments are behaving the same. I think that's what you're saying, and also then using that to control and understand what those mechanisms are doing, and really understanding what that pathway is that's getting to these end results and these biomarkers that we're seeing expressed. And also, my whole thing is, while we do our experiments and we're trying to understand how our models are being impacted by the drugs we test on them, is really having a defined set of positive and negative control drugs. We've looked at clinical data that's been released. We've looked at the way our model performs, and understanding that if we test, for example, TNF alpha is what we use heavily for analyzing like vascular leak and permeability, and that's something we use as a positive control to show that. And using repeated data and repeated tests, we can use that to see that, especially in human vascular cells, that we are getting those end results. We're getting that output when we compare it to unknown drugs and unknown compounds that we're testing.
Sorry. So, some of the answers to your questions, I mean, based on what I understood, is that, with those animal models, I think that, in some instances, depending on, say, if it was, say, something that was a clinical finding, you didn't see it in those animal models. If you do see that in the clinic, going back, I don't think, if there was a human model to go and investigate that mechanism, I think then, you don't need those animal models. Where I see there, that issue again is where you have those, one species shows it, the other species doesn't. How do you know that, what do you think is going to happen in the clinic, and how do you take that, how do you make that prediction? And you have a lot of other data to do that, but all the data that we have, we're still getting, say, clinical liver toxicity, and we can't predict that. So, having those model systems, and I think they really just need to, hopefully in the future at some point, be developed, but to have that confidence, and that you say, okay, if I'm developing a model and I want to use it, say, for predictive toxicity, that I know that model, if that is, it recapitulates, say, based off of, whatever positive and negative controls you have, this model system, it doesn't even all have to be in one, liver model system. I mean, they're all not going to be able to cover every single type of toxicity. They all can't have every single cell type that we want to put in there. I mean, it's just probably not practical in the end to do that. But if you had, a handful of different types of, models, this one has, these types of cells, but we can, recapitulate this type of injury. When you get into the situations where you have differences between those species, it would be good to understand, and, you have confidence in those models, that clinical, that human model is then hopefully that tiebreaker. And I would say in the end, where it comes down to me is that if we do that work and, the modelers build those models and we can do that, is there going to be enough confidence in, the regulatory agencies being able to see that difference and then saying, okay, here's this human model that, we don't see that effect in. Is that going to be enough to push push that forward and be able to move forward with your compound versus saying, okay, well, it's just an in vitro, it's an in vitro model and you still need this fold cover for that, because that's where I think a lot of the compounds fall down, is that, there's a bit of safety cover. You don't understand, where that is. You have two different species. You're basing your, clinical doses off of, the most sensitive species. But if you can prove that perhaps it's not that sensitive, then it's really the understanding from, the right context of use, understanding, how the regulatory agencies are going to respond in those situations when we actually do have these models to do that.
Just speaking from a very personal experience, I've actually successfully used animal MPS before to do risk findings in rat like hyperplasia that we found that was associated with phosphodiesterases, and I find them quite useful actually for the contexts that you discussed here. So I'm glad that people are investigating this area more.
Yeah, Jonathan Jackson from Drug Safety within the global discovery and investigative tox group at Pfizer. I just want to say thank you guys, the presenters, for the very thoughtful presentations, and also the panelists for their thoughtful answers to our questions. But I had a question to the group as to, in the presentations, kind of just looking at some of the characterization that you were suggesting that we should include in our complex in vitro models. What I didn't see was characterization of hepatobiliary disposition, and actually having a 2-compartment liver model where we actually have a distinct bile pocket. Because I think that that's one of the major issues, is that we're not recapitulating a major compartment with the liver that would have downstream effects, and I've noticed that, there's a real lack of understanding. I don't know if it's a lack of understanding or a lack of how to build it, or just maybe a lack of, considering it to be achieved in the next generation complex in vitro model. So I'd like to hear you guys' thoughts on that.
I just want to say, I think that's a very valid point. I've seen MPS systems for gallbladder and hepatocytes, but I've never actually seen them together. So I think that would be actually very good.
Yeah, John, I think you make a great point. I think that there's still further technical development that can be done to build those models. I think there's a lot of things we can use with the models we already have to bridge that gap. I think there's a number of other endpoints, cholestatic injury measures, that we can use to assess bile and the biochanaliculi that form within the hepatic niches that we already have, that can be used right now, and I think could be incorporated into some of those lists that were presented on the slides. And I think that then it can be built upon about whether the specific requirement for that. But I think, again, this was alluded to earlier, I think we also have to balance, and as a developer, we think about this all the time, right? We've got to balance the usability, throughput, scalability, because if any of these models are going to get used in anger and at the scale that we want them to, they've got to be usable and simple. If they're going to have multiple compartments, multiple chambers, like half a dozen cell types, it's going to make the usability really hard. And so we've got to build up to that as people kind of get the confidence to it. But in reality, it should be possible, but it's certainly never been done. I've never even seen a crazy academic build that chip yet, but I don't think it's an impossible task.
But we have to recognize that a model without that compartment is opening up to false negatives.
Yeah, we have to understand where the limitations are, right? Every model's got limitations. Sure. Yeah.
And yeah, that's kind of the trade-off that you see when you going from in vivo animal models to these complex in vitro models, is you go from having a complete system of multiple organs, multiple tissues, very complex systems that are touching in constant interaction both up and downstream. And then as the model builder, it's our job to take those specific use cases and I feel like I'm beating a dead horse here, but that context of use that is the most important, the most applicable into the industry, and find those ways that we can build a model that really addresses that one specific aspect. It's been going from an academic lab and in my research to then coming into industry, right? The trade-off that you get with building these really advanced, complex models that as a, master's student, PhD student, postdoc, whatever, where you can spend and really baby these chips, and you can have these big complex models get these really complex outputs and multi-tissue systems, but then translating them to industry where, a lot bigger the focus is really pinpointing one of those use cases, those high value, high throughput, and finding that balance there to get a model that is duplicative of a human or in vivo system, right? I think that's the way the industry is going to go in the future, especially as more and more multi-organ chips come up, being able to maybe not only combine them as one system, but link these systems together, various ways to emulate that downstream effect and those multiple tissue groups and organ groups actually coming in contact with each other. Like I said, it's where the technology is at currently and where it can go. I think there's, tons and tons of possibilities out there.
Sorry, and I'll just say, yes, in answer to your question as well, along with this question, I think that, we were kind of being very simple in, the slide saying these are the characteristics, and what he said actually prompted me to my one answer to, your one question is that, we need to understand what that, PK and yours as well, we need to understand that. And I would say when, I was presenting, I think that there was one, slide in there that I kind of glanced over, but it had different categories on there. And it was like having the right DMPK, having the right, that kind of information in these systems. And that comes into play in those complex in vitro models. And, I have experience where, we've done some experiments and, we ended up getting some results and we went back and it wasn't really, we were scratching our heads, what does it look like? And we ended up getting the, serum the media from those, we saved it and we collected it and we went back and we assessed, what the drug concentration was in there. And it was like folds off of where we expected it to be. So in developing these models, perhaps, we're not going to have it exactly right, but I think incorporating those types of endpoints into these models and understanding physically what was seen in there. And, you have to pay attention to, do the cells metabolize it, how, the human or how the animals would metabolize it, they may be different. So take, everybody's point, because I think that there's, multiple questions about that same thing, is that is something that needs to be considered. I think about it as that, a lot of the times that's done really, really early. And within that, once you get to first, to the various different stages, you think about, how that's going to get incorporated, but it's important all throughout the drug development process to get that incorporated into those systems.
Yeah, I just wanted to go back maybe to the value of the animal, MPS, and to just say from the perspective of, farm talks in Cedar, we see the value of these models in the area. Actually, there are multiple sort of places where they have value, but one is to increase the confidence in the human systems. I think that by looking at how changing species and changing conditions, how that impacts the endpoints that you're measuring, that is going to provide us more confidence in the behavior of how those human systems kind of, work. You also have a lot more data with animals from in vivo studies that were conducted in multiple species of animals, and a lot of, drug candidates that never made it, and from other kinds of chemicals that have been used in animals, that you could actually develop a lot more, sort of like have a larger data set that you could compare then, and then sort of draw conclusions so that then you can try and interrogate the human-based system. So that's kind of like the area of confidence, which I think, you've mentioned before, is an important one. I certainly don't think that, there's going to be a situation where people are going to be asked in industry to conduct the studies in animal MPS and then in human MPS. That is not, that I don't see that is going to be, a situation that's ever going to come up. There might be situations where an animal MPS though might be good for, in terms of like if you want to select in discovery which species you want to actually do some of your first animal testing in. Some of these animal MPS might be useful in being able to select the proper species for the type of endpoint that you're trying to evaluate. And then also, after you've seen, you've done the animal studies, if you see see a differential toxicity to try and explain that toxicity. So I think it really depends on what the purpose is that you're trying to use the model for, which is, I think we keep going back to this, context of use thing. And that's why it becomes so important to articulate the context of use that's very specific. We can't sort of develop generic contexts of use, but there's value to any type of model and under any type of context of use. I would say that I certainly think people should really invest in developing these animal MPS, because we find their what they contribute to be pretty valuable. So thank you.
Thanks, Nikisa. I would just wanted to give a quick example of, I would say in line with what you were saying, is that for the drug-induced vascular injury working group, we have drug-induced vascular injury, and specifically characterized by medial arterial necrosis, which is the outcome of that. That is something that there are no biomarkers currently for. You can assess that across the various different species. It occurs in some instances in specific vascular beds in rats. In dogs, it can occur in different vascular beds, so the coronary arteries versus mesenteric arteries. It doesn't occur with all compounds. Sometimes it's not associated with changes in cardiovascular parameters. So when you're developing these drugs and you actually see that, say in a rat, or you see that in a dog, you didn't see it across both species. There's actually no human model, I would say right now, that you can even go to and recapitulate that. So what happens is that you need to have, based off of maybe 2 years ago when we had a SOT workshop and, discussed with, the FDA, what we've been saying is that you need a 50-fold, cover to be able to have that. And if you don't have that, we kill compounds because of that. So like in line with what you were saying, like in those situations, what, we're trying to do is develop models in the animal. So starting in the rat, can we develop a model that, really can show you the same endpoints? Now, it doesn't have to have all the factors. I mean, it would be great if it did, but when you went back to, somebody said, applying omics, I think, into that, there were, within GSK, like we developed, a predictive model based off of like administering 28 different compounds to rats. And we have this gene panel that, we're going to assess. So you have to have something that you would, I would say, be able to, assess across those different models that you're developing. So using, say, that gene panel, based on what's out there, maybe you don't have to develop something new. Maybe, that predictive panel, just looking in your model, maybe it'll hit all those endpoints. But, understanding and having that model that you have confidence in the rat, understanding then, you didn't see it in the dog, but you have a model in the dog that you're confident in too. So if you don't see it in both, and then using that information and having a human system that recapitulates, say, the vascular components, and that you have confidence in, but you haven't really tested in that understanding, because we don't know if it's going to happen in humans. But if that human model can respond to different stimuli, can do that, then you kind of have this, I would say, overall system where you can look across the species, and that gives you confidence in the fact that you can move forward because everything's kind of aligning, even though you don't have biomarkers and that. So I don't know, just an example of what you said, I think, a prime example.
I just want to point out that sometimes you can be misled by the animal model. A great example is phenyluridine in the rodent. So, mice and rats are absolutely resistant to the LASH, the lactic acidosis hepatotoxicity effect, which in humans ended up with 4 or 5 liver failures in patients in phase 1, and the drug was killed. And the reason is that the transporter for phenyluridine in the mitochondria, where the damage is done in the DNA replication of the mitochondria, the transporters in the rodent mitochondria are dead, whereas in the humans, they're very active. So you got the effect in humans, but not in the rodents. And that was this is already a number of years ago, but this was a misleading finding. And so more recently, the Stanford group, of course, did the chimeric mouse model, where they put human hepatocytes into the in vivo mouse to get to replicate the toxicity effect of phenyluridine in vivo. Because the mouse hepatocytes don't care about the drug. It's only the human hepatocytes. So it works both ways, and you have to be aware of those potential biological differences.
I just wonder, out of interest, I don't know if the sponsors here are like comfortable informing us, but I wonder if they'd be comfortable letting us know if they're using animal MPS for decision making. I suspect they are, and based on my personal experience, but I wonder if, would you be comfortable to, do you use animal MPS in-house?
So I would say from just, my perspective, I would say, complex in vitro models are used. I would say for the most part, they're used in, that earlier phase of development. I would say we're trying to go towards that, utilizing them, to be able to be, predictive. I think it's a long ways away just based off of the systems that are currently developed. I think it's the, thought that that's where we want to go. Things can be used for, mechanistic insight and those types of things. But I would say it's just, in my perspective, I think the the ability is there, the thought that we can use it for those perspectives is there, in everybody's minds. But it's really being able to characterize those models, test them, and it does take a long time, and understanding once you have a modeler say, oh yes, this is a perfect system, and here's a paper, and predicts everything 100% of the time. It's also then, I would say, and from my personal opinion, there would be saying that, okay, well, I want to see it in my hands because I just want to see it recapitulated. So then, you do all that, methods development in-house, to try to, or, run the studies with the developers to really like prove something before you can actually put your compound. But I would say it's just, my perspective, it's, it's used, people are trying to develop models. And through PSCC and, others, we hear, all folks, talking about the various different models they're trying to develop. But just based off of even, the IQ survey that was done maybe 2 years ago now, and there was one earlier than that, nobody's actually taking those and submitting them. But I would have to say, from my knowledge across the industry, people are trying to use them. They're just, I would say, more hesitant to put them in, submissions, just with the uncertainty of how the regulators will respond to the data that's getting put in there.
This is a question for Kevin. Would it make sense, because we're talking about, drug metabolism and differences in detoxification? So take animals, for example, they have carboxylesterases. We're far fewer on that side. And then, it's not just P450 monooxygenases, it's monominoxygenases, aldehyde, you name it. So would it make sense then, because you're already picking up that group of chemicals that you want to test, to kind of focus and be mindful about it? There are clear differences, right? And they go beyond to what the IQ prepared to kind of accommodate for that, and we can really see that distinction. So if there is a gap, and animals are able to detoxify something, then we're clearly missing something in clinical trials. So why not just focus on drug metabolism? Phase 1, phase 2, and phase 3.
So just to give you a preview of the drugs that are coming your way, some of them do actually contain like ester groups, for CS, and other, structurally available hydroxy groups for glucuronidation and things. So we did take that into consideration, the group in DARS. So I think I would love to get your feedback actually once we roll it out.
Which is actually a nice example, because spirit is more toxic, right? So if you
Right.
test in mitochondria, and you're going to see that you're going to get a lower IC50 than the whole cell. Because if you take metabolism into account, you stop 3 or 4 stops, and it's not going to go further.
Right.
So that one is actually interesting.
Yeah. Look forward to getting your feedback on that. Thank you.
Kevin, I was going to ask you a question. Does your list, is it all small molecules? And do you think that's appropriate?
It is all small molecules, yeah, just because that's my comfort space, being a chemist. So no time for large molecules. But I think we will just consider maybe large molecules later, or oligonucleotides or MABs or whatever. But yeah, all straight up small molecules that are commercially available and well known.
I wanted to ask another question that's sort of building off of one of Mark's comments. So this gets at potential interspecies differences of animals, humans, between species. And so like, Mark, like you pointed out, it can go both ways. You miss something in the rat or the human or whatever species. I don't even remember what species you were talking about. But, so a question for the sort of field of DILI experts is and with the liver, it's also, transporters and metabolism differences between species. But do we understand the differences between species? And are there, like there's always going to be differences, and a model is never going to be a human of any kind. But like, are there some critical, failures that we're concerned about? Like, and then the question is that like it's a difference in transporter, a specific transporter function, and so is there another in vitro test, let's say, or we're already assessing if a drug is a substrate of a certain transporter or metabolized by a certain CYP enzyme. And so you can, de-risk, answer the question differently. It's something to consider and to think about, beyond this discussion also. But I don't know if anyone has any thoughts.
So there are functional assays to determine, if the target that you suspect is causing the toxicity is in fact, the target. But the tough part is identifying that in the first place. That's the challenge. When you do these early stages, you're just rooting through enzymes and ion transporters and all sorts of things. So it's really hard to pinpoint it down. But when you do, there are plenty of CROs that can do confirmatory studies. You can do it in-house, I guess.
Coming up, I think, based on characterizing those models and, the example that was given where, it didn't recapitulate and, it wasn't exactly, if the rat wasn't the same as the human, it didn't have, specific, the transporter or gene. And that's kind of where we have been talking about this morning, is understanding and how do you characterize those models. So depending on that context of use, and I'll kind of bring it back there, is that if you have a well-defined context of use and that you're looking for, to understand X for this particular compound, there's a lot of work that gets done like very early that's characterizing, say, mouse models. Like, does it have the target? Does it have the pathway of interest? So that's more getting to that pharmacology. And I think that, these models also need to be able to, maybe they can't do that. Maybe there is something that is different, but I think that's where it is understanding, the model that you're going to use and understanding the MPS that you're going to use. Because if, say, yes, the rat doesn't have, a specific, pathway or something, similar to, say, the human, then that's not a model that, you would want to, choose if, your target is expected to do something, and maybe that, it is something of importance. So I think it's really, the model developers understanding and characterizing their models very thoroughly, so that if you say, somebody would say with omics, take that, my opinion anyway, personal opinion is, take that model, understand what's expressed in that model, what's expressed in those cells, what's expressed if you have a model that has, different cell types, because that could exchange, it's not just those individual components, it's understanding those individual components, putting them together, understanding how they behave when they're all together. And it's just really as a company or somebody else developing that, looking at all that data and making sure you pick the right MPS model or any other model, like you said, and like you said, maybe there's something better that can address the question that you have.
And I think this was sort of brought up earlier, right, is that we've got to view the mouse or the rat or the dog in the same way that we view the MPS. Like, the same scrutiny has got to be given to every model, because every model is a model. They have different characteristics, they've got different flaws, they've got different pros, different cons. So we have a standardized framework by looking at any model, and actually it will raise up the quality of data that comes out of any system. Same for computational models as well.
And
Yeah, no, I just about follow up on what Deirdre's saying. I think we all gravitate towards markers, biomarkers, endpoints, and things like that. But I think just as much effort needs to go in up front. And so just like what you're saying, what do the cells express? How does it change when you start putting multiple cells together? I think those are lists that we need to think about, so it's easy to say, oh yeah, I'm going to take a Kupffer cell and a stellate cell and a hepatocyte, put them together, and now I have a liver. But are those, even across species, are they expressing key markers that we all want to have? And so do we need to put a list of those marker enzymes, the different cytochrome P450s, the different transporters that a dog hepatocyte or a rat hepatocyte or a human hepatocyte, whether it's be from an IPS or a primary, should they be expressing X, Y, and Z before we're going to go in and start evaluating compounds? So I'd like to hear what your thoughts are in terms of creating that sort of standardized list up front.
I think in principle that's a great strategy, right? But for the point that Kevin made, the number of variables that are out there are enormous, right? You take one of those cell types as an example, like if you take primary human Kupffer cells, you go out onto the commercial market and take 5 different vendors of primary Kupffer cells, they're all isolated in different ways. They're all prepared in different ways. So when they come into our lab, right, there's a whole bunch of variability. So actually some of that, I agree with you in that sense. We get some standardized ways to evaluate individual cell types so that when they go into any of these chips or any of these models, whether it's a spheroid or an advanced MPS, at least there can be some basic starting point. And then you could apply that beyond human, right? Then I think the human are therefore the most advanced. Let's start there, and then we can build into some of these other species.
I just think people don't think about that 5 different sources of cells are different. They say, oh, I'm taking a Kupffer cell that's commercially available, and they don't sit there and start thinking about that variability inherent in something that they think is commercially standardized. Yeah,
I completely agree. And no one's asking that question about, and I think that's what this group have got to start the community talking about. Yeah.
Good afternoon. Thank you for a wonderful talk and discussion. I have a quick question. So just I'm wondering, once we identify appropriate CIVM, we are talking about the validation. Have you would like to seek your thoughts on what do you validate and against what you compare? How do you prove that your system is working? Is it just reproducibility check or what else? What your thoughts are?
I think the ultimate evaluation is, can you recapitulate the findings in the clinical study? I think if you can in your in vitro system, then I think that's a good way along to validation.
I think also in terms of the validation, these complex in vitro models, they're being built and developed for a specific use case and have specific outputs. Most of the time, they're not a cure-all solution, these NPS, to be directly translatable into the clinical model. And so really narrowing or being very specific and very clear with what the outputs are of these models, and basically validating and treating it like any other assay that would come off the shelf or be used in these experiments to build your portfolio to ultimately move into a clinical trial, or support your data to know that's safe enough to move into a clinical trial. But validating around what those very clear context of uses are, what those clear designed outputs are going to be, and like you said, continue repeatability, recapitulation into a clinical model or into other animal models that we're doing NPS with animal cells, and repeatability across several experiments, several different users, those aspects, and making sure once again you're hitting those desired and defined outputs with these models.
I think the only thing I'd add to that is that the quality of clinical data to compare against is sketchy at best. So I think it would be enormously valuable for some sort of community consortium-based effort to bring together some clinical data for a particular context of use, where actually it was properly compiled, it was properly standardized, that then would allow some of these models, that there would be a lot of benefit to go back and forward there, because there's what's out there and what's published is limited at best and not standard in any way.
I'll just, make a comment that, with the, CPATH and, with PSCC and understanding, your question that you asked, I would say that there are, and I don't know, Nick, if you want to, say anything, but I'm put you on the spot, but I would say that, that information, is trying to get pulled together in a biorepository that, hopefully, will be accessible. But I mean, you know more about that, so sorry for calling you out.
No, I'm happy to answer. In terms of data, we do have we're calling it the biomarker data repository right now, and we're focused initially on kidney injury, but expanding into other tissue and organ injuries in the future. And the goal is to have primarily clinical trials that are well characterized and complete in terms of demographics, the assays that were used, and description of those assays assessed, as well as the standard clinical chemistry, and use that for pursuing future qualifications is actually part of the goal there and objective. But there are definitely other ways that we can interrogate that data. And from data within PSTC, there have been a few nice publications looking at some of the rat data and applying new algorithms for preclinical assessments that we've had that came out from one of our co-chairs. And so definitely some of the data that we have can be reinterrogated in different ways from within the membership, and has led to some very nice publications, and something we would be happy to look at ways where we could make this something that would apply across both looking at the clinical trials that we have in hand, as well as comparing that to studies run in complex in vitro models.
Okay, so we're at 3:20 or a little bit past. Thank you to the panel for your input here. This was great. I think we're going to take a short break, so everybody can grab some caffeine and come back for the session number 3. So again, thank you, and